Hello, dear students. In this video, we're going to continue what we have started before in our digestion chapter. In this video, we're going to talk about activity 3 about the enzymes, the agents of digestion. We're going to identify the favorable conditions for the normal functioning of enzymes. Also, we are going to study the pathway of the nutrients, and also we're going to identify the route of nutrients inside our body and exactly inside our digestive system. We're going to talk about chapter 1, activity 3, enzymes, the agents of digestion. What are the conditions needed for the proper functioning of the enzyme? To understand this, we're going to move to an application on page 2021. We're going to interpret the results of the experiments shown in paragraph 1 on pages 2021 and also to deduce the enzymatic property verified in each of the performed experiments. Here we have two documents, A and B. Document A is showing the experiment to study the property of an enzyme, which is called specificity, or the meaning of specific. Here we have two test tubes, A and B. Both of them, they are placed in a water bath at temperature 37 degrees Celsius. In tube A, we added cooked starch and fresh saliva. In tube B, we added an albumin of an egg, which is protein, with fresh saliva. So both of them, they have the same condition, instead, but we have different substrate. Here we have starch as a substrate, while in tube B, we have albumin of an egg as a substrate. After 1 hour, we move after 1 hour. So after 1 hour, we have in Fehling test in tube A, it gives brick red precipitate, meaning that the starch is converted or is transformed or digested into reducing sugar. While in tube B, with violet test, it gives a violet color, meaning that the protein is still found in tube B, meaning that there is no digestion. So after adding fresh saliva to the cooked starch in tube A, the result is brick red precipitate with Fehling test. What does this indicate? This indicates the presence of reducing sugar obtained from the transformation of starch in the presence of fresh saliva. So saliva converted the starch into reducing sugar, and to be more specific, the enzyme inside the saliva, which is called salivary amylase, works on starch and converted or digested into reducing sugar. While after adding fresh saliva to albumin in tube B, the result is violet color with violet test. This indicates the presence of proteins and or polypeptides present in the albumin and not transformed by the fresh saliva. Now let's move to the enzymatic property deduced here. Here we deduce the enzymatic property is every enzyme acts on a specific substrate. Every enzyme works on a specific substrate. In this case, enzymes that in saliva could digest starch into reducing sugar. That's why we have a positive result with the Fehling test. But this enzyme that is present inside saliva, it cannot digest albumin. It cannot digest the albumin, and it still persists and gives a positive result with a violet. Let's move now to figure C about the activity of the enzyme and the pH in the medium. We know that neutral, the pH is equal to 7. Acidic pH, it's less than 7. And basic pH, it's greater than 7. So here we have pepsin. Pepsin, the activity of pepsin reaches its maximum at the pH equal to approximately. So the pH equal to, it was maximum. While salivary amylase, its work was maximum at the pH approximately 6.5, 6.7. This is the maximum pH, is around 7. So it's a neutral. While trypsin, it works best at the temperature at the pH about 8.5, which is basic pH. So here we can deduce that for pepsin, For the pepsin enzyme, the pH increases to reach its maximum at the pH approximately 2, and then it decreases to reach null or back to its minimum when it increases. Also, when the pH was 1, so the activity was minimum. Here, salivary amylase, when the pH was 5, the activity was minimum. As it increases to reach about 6.5 or 6.8, so this is the maximum activity of the salivary amylase, and then this activity decreases as the pH increases to 9 to go back to the minimum. While this trypsin, its enzyme was minimum at the pH of 6.2 or 6.3, and this activity increases to reach its maximum at the pH equal 8.5, and then as pH increases, the activity of the trypsin decreases to reach its minimum again at the pH of 9.5 approximately. So for pepsin enzyme, as I said, for pH equal 1, it's acidic medium. The speed of the reaction is minimum with this enzyme. So as pH increases from 1 to 2, the speed increases from minimum to maximum. It reaches the maximum value at the pH equal to. Also, as pH increases from 2 to approximately 3.8, the speed decreases back to its minimum. So what's the indication here? This indicates that an increase in the pH, it stimulates an increase in the speed of the reaction, but up to a certain limit, which is pH equal 2. Above pH equal 2, the speed increases. The speed increases from minimum to maximum. Above pH 2 and reaching 3.8, the speed decreases back to its minimum. So the increase in the pH stimulates an increase in the reaction of pepsin enzyme up to a certain limit. So above 2, the enzyme will go back to its minimum level. Okay, so pepsin enzyme, you have to keep it in mind. Pepsin, it works in acidic medium like Pepsi, it's acidic. So pepsin, it works in acidic medium. For salivary amylase enzyme, salivary amylase is found inside the saliva, inside the mouth. The medium is neutral, so it's approximately 7. So for pH equal 5, which is acidic. Why it's acidic? Because less than 7. The speed of the reaction is minimal. As the pH increases from 5 to 6.5, here is 6.5, we record it as a neutral one. It's 6.5, it's around neutral, it's around 7. So the speed increases from minimum to reach its maximum. And as the pH value increases from 6.5 to approximately 8.5 or 9, this speed, which is the speed of the reaction, decreases back to its minimum. What's the indication here? This indicates that an increase in the pH stimulates an increase in the speed of the reaction of the salivary amylase enzyme up to a certain limit, which is 6.5. So above 6.5, the speed of the reaction will decrease back to its minimum. And below, the speed of the reaction also its minimum. So salivary amylase, best optimum pH, it's around neutral. It's around 7 pH. So here, till now, the variable factor we're talking about is only the pH. So the pH for pepsin, it's acidic, while the pH for salivary amylase, it's neutral. pH equal approximately 7, around neutral. So as you see here, the enzyme works at specific pH medium. For trypsin enzyme, the pH was the reaction for trypsin enzyme, the pH about 6, which is acidic. The speed of the reaction is minimum. So as the pH increases from 6 to around 8.5, the speed increases from minimum to its maximum. So maximum at 8.5, which is basic. And as the pH increases from 8.5 to approximately 10, this speed decreases back to its minimum. What does this indicate? This indicates that an increase in the pH stimulates an increase in the speed of the reaction of the trypsin enzyme up to a certain limit, which is 8.5. So trypsin works best and optimum pH, it's basic 8.5. So enzymatic property deduced here that every enzyme needs a proper pH medium, or it needs a proper chemical medium in order to act. So we have till now two properties for the enzyme. Every enzyme works at a specific substrate. Every enzyme works at a specific pH medium or a specific chemical medium. Let's move now to document D, action of the enzyme and the temperature. If you see here, the activity of the enzyme was 0 when the temperature was 0. And as the temperature increases, the activity of the enzyme increases to reach its maximum 100 degrees, where at temperature approximately 37 degrees Celsius, which is the body's temperature. And as the temperature increases from 37 to 60, the activity of the enzyme decreases back to 0. So here the variable factor is the temperature. So for temperature equals 0, the activity of the enzyme is 0. As temperature increases from 0 to 37, the activity of the enzyme increases from 0 to 100, which is the maximum value. As temperature increases from 37 to 60, the activity decreases back to 0. This indicates that increase in temperature causes or stimulates or provokes an increase in the activity of the enzyme up to a certain limit, which is 37 degrees Celsius. So here we have to deduce that enzymatic property deduced every enzyme need a proper temperature to act, which is the best temperature, 37 degrees Celsius. To keep in mind, you have to know that optimum temperature in our case study is the 37 degrees Celsius, since the enzymatic activity and the function of the enzyme is at its best at temperature 37 degrees Celsius. 2. Inactive temperature in this case is 0. Why? Because the enzymes are inactive. So inactive temperature, because the enzymes are inactive, they are in sleeping mode, their activity is 0. 60 degrees is the denaturation or the destruction temperature. Why? Because the enzymes are destroyed or they are denatured under high temperature and they cannot act anymore, even though we bring them back to temperature 37. While in the inactive, they are sleeping. When we give them temperature 37, they will work again. This is the difference between denaturation temperature and the inactive temperature. So denaturation temperature, it's the enzyme is destroyed, it cannot act anymore, while inactive, the temperature can work again when the temperature becomes 37 degrees. So to keep in mind, optimum temperature is 37, so optimum is 37. Why? Because the enzymatic activity is at its best at this temperature. Inactive temperature in this case is 0, since the enzymes are inactive at this temperature. When we give them back to temperature 37, they will work. 60 degrees in this case is called denaturation or the destruction temperature. Why? Because the enzymes are destroyed or they are called denatured. So you have to know both words, destroyed or denatured at under high temperature, so they can no more act. Their activity become 0 and they are destroyed. If an enzyme is inactive at cold temperature, this is the note, and it's replaced at normal temperature with 37, it becomes functional. Once again, it can work. But if an enzyme is placed at a very high temperature and then replaced at normal temperature, it cannot function because the temperature destroy or denature the enzyme. Activity 4, from food to nutrients. Is it easier for digestive juices to digest small pieces or large pieces? Sure, to digest small pieces. You have to know that digestion is classified either mechanical or chemical. What is mechanical and what is chemical, and what is the difference between these two? Mechanical digestion, it breaks down food into smaller pieces. Also, it keeps the food moving through the long digestive tube. The organs of the digestive system where mechanical digestion occur. In the mouth, we have something called chewing. the stomach, we have something called churning. In the small intestine, we have something called peristaltic movement. You have to know this. Chemical digestion. It involves the digestion of food by the help of digestive juices. So these two juices, they are needed in the process of digestion. Breaks down food into simple nutrients, and it occurs in the mouth, in the stomach, and the small intestine. How are these two types of digestion needed? They are needed by the method that says mechanical digestion of food. So whenever we eat food, we have to chew it. So by chewing the food becomes smaller in pieces. So these small pieces, they allow the functioning of the juices. For example, in the mouth, we have the digestive juice, which is called saliva, that's secreted by the salivary gland. So saliva helps in the process of digestion. So mechanical digestion in the mouth, we have chewing. In the stomach, we have churning, and the small intestine, we have peristaltic movement. So these mechanical digestion, chewing, churning, and the peristaltic movement, all of them, they facilitate the chemical digestion, meaning that they facilitate the action of the enzymes and allow the digestion to finish faster. That's why small pieces of food, they are digested faster than large pieces, because we have mechanical digestion. So mechanical digestion facilitate, it makes easier for the chemical digestion and allow the digestion to finish faster. For example, if you eat large piece of meat and small piece of meat, which one is going to be digested faster? Sure, the small piece of meat. So mechanical digestion has an important role, since mechanical, it breaks down food into smaller pieces. So breaking the food into smaller pieces will help the chemical digestion and allows the digestion to finish faster. Without mechanical digestion, we will have problems in the digestion of food. So activity 5 for chapter 1, the root of nutrients. You have to know the characteristics that favor the intestinal absorption, the absorption at the level of the intestine. We have thin wall of intestinal cells. The intestinal cells, they form a thin wall, large surface for the exchange. That's why it makes a good place for intestinal absorption. Highly vascularized, meaning that rich in blood vessels and lymphatic vessels. Highly vascularized and moisture. These 4 characteristics, you have to know them. Thin wall of intestinal cell. Number 2, large surface for exchange. Number 3, highly vascularized. And number 4 is moisture. For example, in any quiz, I'm going to ask, what are the characteristics that favor the intestinal absorption? You have to say thin wall of intestinal cell, large surface of exchange, highly vascularized, rich in blood and lymphatic vessels, and moisture. So these are the characteristics for the intestinal absorption. You have to know that the routes of the absorption either through the blood or through the lymph. Blood absorbs all the nutrients except fatty acid and the glycerol. These fatty acid and glycerol, they are absorbed by the lymph. So the lymph absorbs fatty acid and glycerol. So the route either for blood or for the lymph. Blood absorbs all the nutrients except fatty acid and glycerol, while the lymph absorbs fatty acids and glycerol. So as a note, you have to know the difference between chemical and mechanical digestion. Chemical and mechanical. Mechanical breaks down food into smaller pieces. Chemical is for the help of the enzyme. You have to know the 3 properties for the enzymatic functioning, or the enzymatic properties. First one, every enzyme works on a specific substrate. Every enzyme works on a specific pH medium. For example, salivary amylase, it works only on starch and doesn't work on protein. This is every enzyme works on a specific substrate. Every enzyme works in a pH medium. For example, pepsin, it works in acidic medium and not in basic. If it's placed in basic, it will not work. For every enzyme works on a specific temperature. Mostly all the enzymes, they work at a temperature of 37 degrees Celsius. If the temperature is greater than 60, for example, the enzyme is going to be denatured or destroyed and cannot function anymore, even though we place them at temperature 37 again. But if they are placed at a very low temperature, which is 0 degree, the enzyme will function again if it's placed at 37, because they are inactive, they are in sleeping mode. These are the notes for our digestion chapter. By this, we'll finish all the chapter, and thank you all for watching this video. Review all the notes, study them, and see you then in another video very soon. See you guys. Bye bye.